So I wanted to get into the soil. The next hour was supposed to be soil, the plant stomach. Now, why the plant stomach? This is interesting. Remember, we were talking about the biology of plants, and we looked at the root system, and we looked at the vascular system, but when we look at plants, we notice that they don't have a stomach, they don't have a large intestine, they don't have a small intestine, they don't have a colon, they don't have all these other intestinal things, but how did God design the digestive system for plants? And it's really interesting, because essentially the plant stomach is the soil. Now, if we think about our own digestive system, we can excrete as animals, well, I don't consider myself an animal, but they classify us in the biological community as animals. But either way, we can excrete certain acids and a few enzymes and things into our small and large intestines and try to break down things. But really, the majority of the digestion in our small and large intestine is through microorganisms, bacteria particularly. This bacteria is capable of excreting different acids, different enzymes to break down different biological processes and carbons, and taking nutrients that otherwise would not be available to us and making them available to us so that we can digest our food. Our human digestive system, as well as the digestive system of various animals, is very much dependent on the bacteria that is in our digestive organs. It's not that different for the soil. But the interesting thing to note, in the medical community, you don't get a lot of talk about how your diet affects the microorganisms in your gut. But I can't give you every single name. In fact, the scientific community right now, whoa, what happened? I'm sorry. The scientific community right now for soil, or for biology, or I'm sorry, bacteria and fungus, there's so many organisms out there, the number, they don't even know the number. It's in the trillions and trillions of different organisms. And because it's so diverse, what they do is that they have decided to stop naming it the way that they normally name it. So they're not calling them just bacillus this, or azotobacter that, or et cetera, and giving it all these different genuses and families. What they have decided to do for soil science is get away from that whole naming that the rest of the biology science discipline uses, and go to simply just 16S RNA sequencing, which is taking a certain portion of the DNA code for bacteria and naming them based off of the deviations in the genetic code of bacteria, and then the same thing with fungus, except they use the 18S DNA sequence for fungus and the 16S DNA sequence for bacteria. It's so complex, it's so diverse, that they themselves don't even know how to name it. That's how crazy it is. That we as mankind, even the smartest people out there working on this thing, have no idea what to call these organisms. So they just got away from the stereotypical method of doing things. So when you start looking at genetic diversity, so now everything is about genetic diversity and not trying to identify one particular species or a different particular species. So when we start looking at the genes, what we're doing now is we start comparing the genetic diversity of bacteria that we find in any particular soil. And then we start thinking of the conditions that the soil needs to be in order to see these deviations in genetic diversity. And what they have discovered simply is that there is a significant amount of change in the percentages of certain genes based off soil conditions. So, an example is, when we look at soils that are confined feedlots, and we start looking at the percentage of the bacteria that you find in there that fall under the category as acidophiles, which means that they survive in high acid environments, which is what? Our digestive system, right? Our digestive system is a high acid environment. Most of our organisms here should be acidophiles. So when they look at that, what they discover is that the soil has huge percentages of acidophiles. If you go to the forest, you find that a lot more fungus there. And if you go to agricultural fields, you find less fungus, less bacteria, but then you find some other species that are not so favorable. So what they start noticing is that really it's all about what you're doing with the soil. If you want to know what percentage of bacteria or genetics are in your soil, you have to start looking at land use management. What are you doing with that particular piece of land? So when we start to consider management and applying certain inoculants that you find, you really need to consider what are you putting down, and are you really going to make adjustments in that soil based off the inoculant, or is the environment in that soil so different that no matter what you inoculate it with, you're just going to kill them all? And I'll give you an example that is not microscopic. I'll say, okay, we as human beings, what is it, 65, 68, whatever in this room. Imagine you cook all of us, just like we are right now. Basic coats, basic sweaters, and you dumped all of us in Antarctica. How long would we survive? We wouldn't survive very long, would we? So it's the same idea with inoculants. If you do not change the environment in that soil, and you go out and you spend all this money in inoculants and you dump them in there, what's going to happen? You're just going to kill them all. It's all that ever happens. So you get on these websites and they make real big promises, and when you buy these things and you put them, oftentimes it doesn't work. Why? You got to change the environment. Well, what do? Well, this class is not going to necessarily discuss on what to do. I think that other people here have already discussed quite heavily on what to do to change the environment in that soil. But essentially, you got to get the chemistry balanced. When you get the chemistry in your soil right, your biology will follow. Because when you get the chemistry right, you get the environment right. And when you get the environment right, the bacterial organisms that thrive in that environment will thrive. And the ones that don't thrive will not thrive. They'll die off, and there'll be less of them there. But that would be my suggestion to do that. Now, we're going to get into the soil food web. Now, when we start speaking on phosphorus, I'll talk a little bit about the other anion exchange capacity. And what I mean by that is we hear a lot about cation exchange capacity. It's been talked a lot at these conferences. I'm going to talk a little bit about anion exchange capacity and what William Albright considered to be anion exchange capacity. And that essentially is the soil food web. Now, what is that? The soil food web. Has anybody ever heard of that term before? Okay, great. I was scared there was going to be no hands. I got maybe half a dozen hands. That's great. So the soil food web is essentially the cycling of carbon through the soil. What does that mean? Anything that ever used to be alive. Anything, including us. We all go to the dirt. Ashes to ashes, dust to dust, is what the Bible says. From the soil we came, to the soil we shall return, was a curse that God gave to Adam, subsequently to each and every one of us. It is no different for every single piece of biological life on here. It's all going to eventually die. But at some point, it was all alive. So when all of this looks at from the small animal bird being up there pretty high, trophic level, all the way down to your bacteria, your protozoa, and your fungi, and how exactly these nutrients move. When we get back in the next hour, I'm going to talk about the soil food web and a few of the other nutrients, and we're going to take off from there. Thank you. Okay. All right. I think that's good for the recording. Soil, the plant stomach. We talked about this a little bit the last hour. I want to get into the soil food web, but before I get into that, I wanted to share with you, I think most of you know who William Albright is, and a gentleman that wrote a number of books. His name's Charles Walters. I'm quoting the book Eco Farming, page, let me see, I've got it in here, page 213. It's written by Charles Walters, and in that, he's quoting Albright. So I'm going to quote Walters quoting Albright. A lot of quoting here. Okay, so it says, this is on the question of phosphorus. He was being asked the question, how do we, since phosphorus, the particular species of phosphorus that the plants want is referred to as orthophosphate, which is a P2SO4. And the question was, how do we manage phosphorus? How do we measure for this? How do we concern ourselves with phosphorus? And this is the answer that was given. Again, so he starts off by saying, this is Walter saying, we ran this up the flagpole with William A. Albright one day while a tape recorder was running. His answer exhibited the usual patience as he repeated what he had gone over a few dozen times before. Only this time he made the phosphorus connection. In the soil management, he said, we associate the inorganic nutrients, calcium, magnesium, potassium, sodium, iron, aluminum, zinc, copper, cobalt, manganese, and others with positive ionic characteristics, with the soil's colloid humus clay fraction of the opposite, negative electrical character. The essential nutrient elements, namely nitrogen, sulfur, phosphorus, boron, chlorine, iodine, molybdenum, etc., and others, are empirically associated with the organic matter of the soil. So why not envision these anions or negative elements corrected with decay stages of organic matter when a wider carbon-nutrient ratio reserves as a source of energy by the carbon first to the fungi, then to the bacteria in the soil? Why not visualize the negative elements as serving simultaneously in plant nutrition as chelated and large molecular complexes of microbial origin? Xin-Chen Chang made the point in 1931 that considerable phosphorus ranging from 0.1% to 0.5% reached the soil in organic materials. Release of this phosphorus depends on decomposition of organic compounds by microorganisms. It is for this reason that manures, green or barnyard, or compost refinements thereof should not be valued alone for their nitrogen. This statement was made in the early 40s, and it was just in the last 10 to 20 years that people are getting intelligent about compost and the effects that it has on the buildup of phosphorus in the soil. But the more important thing that I want to stress out or bring out and talk about is the soil food web, which is essentially the breaking down, metabolizing, consuming of anything that was ever alive, whether it's a dead person, a dead animal, a dead plant, a dead worm, a dead bacteria, fungus, etc., etc. All of that composes part of the soil food web. Ashes to ashes, dust to dust is what the Bible says. We were all once, from the dirt we were made, everything, now to the dirt we shall return. When we're in that process of decay, those nutrients that were necessary to build up that organic life are slowly released through the metabolizing of soil organisms. Whether it's bacteria, whether it's fungus, whether it's earthworms, whether it's protozoa, or little rodents, or arthropods, or springtails, or snakes, or birds, or the big mean lion, or us, human beings, whatever it is, when we break down, when we are decaying, when there is some level of decay, there's also, along with it, the releasing of nutrients. And so the question was then asked, and this is why I say I prepared this as advanced level, is how do we then manage for phosphorus? How do we measure phosphorus? We really need to look at our organic matter because that's where phosphorus is hidden. I shared earlier how phosphorus will combine with calcium to form bonds that are not broken by any type of plant exudate or even bacterial. Except for one species of Bacillus metallitis, I think is what it's called, that can actually break down phosphorus. And then otherwise it's fungally. It happens through fungus. And some of its exudates can break it down. And I have some images I'll show you in the 6th hour of this presentation with respect to basalt and other minerals that were taken recently at Oregon State University with a helium electron microscope, and it shows exactly the kind of weathering that fungal hyphae puts on basalt and other minerals that form in the soil. But for now, what I really want to share with you is that the bulk of that phosphorus is coming from your organic matter. So as you study soils, you keep hearing that word, OM, organic matter, organic matter. Folks, there's some chairs up front if you'd like to take a seat. So we really need to, it seems like with soil science and with improving soils that it all goes back to organic matter. And this is why so many people jumped on the compost bandwagon, including some of the early reformers of agriculture, if you want to call them that. Even, what were their names? Rodell, even Steiner hopped on that bandwagon. He added a lot of hocus pocus to it with some of his spiritual applications that he wanted to make and his bullhorns and other funky stuff. But the real thing, the real magic that was being done there was the fact that he was recycling nutrients. He was bringing in organic matter. None of the other occultism really favored him at all. Let's see, who was the other one? I forget the names, but anyhow, yes, those early pioneers of what eventually became the organic agricultural movement we're responsible for bringing in that organic matter and understanding the importance of nutrient cycling. Phosphorus is very much necessary for any type of living organism. It regulates the energy through ATP, phosphosynthates, photosynthates. It is required. I showed you the image in the previous hour of the phospholipid bilayer, which is what forms the membrane of every single living cell. Doesn't matter what it is, human, plant, animal, whatever it is. It requires phosphorus to form those membranes, and if it's not available, you're not going to have that phosphorus. So if you got a plant, you got some kind of phosphorus. You can say a plant is phosphorus deficient, but you can never say a plant doesn't have any phosphorus, because if it didn't have any phosphorus, it wouldn't be a plant. Plain and simple. So phosphorus is definitely there. For this hour, what I'd like to focus on is the soil food web. There's this image you guys have been staring at for a little while. Here's another image. We have our organic debris. You see how earthworms will, another word for organic debris in the soil science community is called detritus. That's a new word for you. You can write it down if you like. Detritus is anything that was once alive, like leaves. If you go out and you get a whole bunch of leaves, you put that on the ground, it's referred to as detritus. That can be consumed by earthworms. It can be consumed by fungus. It can be consumed actinomycetes, bacteria, protozoa. Consequently, as well as like a food chain, protozoa will consume bacteria, and then beetles, mites, and springtails will consume fungus. Ants will consume some of these smaller organisms. Earthworms can be killed by, this one actually doesn't have nematodes in it, I think. This picture doesn't have nematodes in it, but you got flatworms, you got rogue beetles, you got centipedes, mites, pseudoscorpions, ground beetles, all these creepy crawlies you see in the soil. They all have a certain purpose that God designed for them, and they're all part of essentially of the food chain of the soil and breaking down organic matter and making it available to nutrients. Every single one of these organisms consumes something and excretes something, whether it's bacteria and fungus just excreting enzymes and acids, or if it's the frass of all these small, frass is a fancy word for manure, for all these insects. Here's another image of 1 square meter of soil. On average, you'll find one vertebrate, which means a bird or a snake or something of that nature. It has, on average, 100 snails and slugs, about 3,000 potworms and earthworms, about 5,000 insects, myropods, spiders, diplurons, about 10,000 rotifers and tardigrades, about 50,000 springtails, about 100,000 mites, about 5 million nematodes, about what's that number? 10 billion protozoa and 10,000 trillion bacteria and actinomycetes. That's in 1 square meter of forest floor. That's an awful lot of life. I mean, if you just stare at that and think about 1 square meter, that's about 9 square feet roughly, maybe 10 square feet of a forest floor has that much biological life put in there by our Creator. That was not by design. I don't think you can study soil science and in any way, in any facet, come to the conclusion that the world just evolved. I don't understand how they come to that conclusion. But when you see that much life, and the thing is that if you remove even just any one of these layers, you really disrupt the soil. If you remove the bottom layer especially. Without bacteria, if somehow you can manage to get in there, and these are some practices that they use now to go in and fumigate soils. They'll treat them with steam or other real strong chemicals, and they just want to kill everything. I forget the name of the product that just recently came out, and it's supposed to kill everything, and I mean everything in the soil. I forget the name of that product. I wish I could remember what it was. They're doing trials with it right now in Oregon. It's supposed to kill everything. Why? Because they don't know what else to use. Glyphosate's not working like it used to anymore, and a lot of these other things aren't working like they used to, so they come up with new, harder, harsher chemicals. So here's another image. We're going to get into soil microhabitats now. We're looking at the drillosphere. This is the point, usually the top 3 feet or so of the soil, where you see your earthworms and a few of the other critters that would drill through the soil. Then you have your porosphere. This is the actual micropores where moisture moves through. When the rain falls or when irrigation comes through, you have these pores that go through the soil. You have the tritosphere. This is the actual, we'll call it the O horizon, but some of you guys don't know what that means, but that's the top layer of the surface of the soil with essentially leaves and wood chips and any of that type of crud that you tend to see in the forest floor. You have aggregate sphere, which is the actual soil aggregates. What forms these aggregates? You can look at soil, you pick it up, you dig it out of the ground, you can smell it. You guys ever smell compost? You know, that rich smell. What is that smell that you smell in compost? Those are the exudates of actinomycetes that you're smelling. And that's that good smell that we like, as opposed to the putrefying, nasty smell that makes you go, ugh, and you want to close your trash can, or you want to walk away from a pile of dung or something. Then you have the rhizosphere. This is your actual roots. You have the rhizosphere is the environment immediately around the soil roots, and the rhizoplane would be the actual right on top of the epidermal cells of a root and its root hairs. We're going to focus on the rhizosphere because I took you from the leaf all the way through the biology, going all the way down to the root system. Now we're at the rhizosphere. And we want to see what's going on here. Now, we've got to keep in mind that this entire presentation is about how do we get healthy plants, sap, so that we can get healthy sap, right? And if we can get healthy sap, because the life of the flesh is in the blood, if we take that principle, we want to make healthy plants. And if we can make those healthy plants, then those natural remedies that so many of us are becoming accustomed to, that we enjoy, will be much more effective if they come from healthy plants and healthy tissues, which requires healthy sap. Where does that sap come from? It comes from the transpiration of the plant. Where does that come from? It comes from the soil solution. What is the soil solution? It is that moisture immediately around the rhizosphere. And what dictates what nutrients are going to be available to the different plant species in that rhizosphere is something that becomes very critical. We've talked about, I think a lot of folks have talked already about chemistry. We balance that chemistry that gets those nutrients balanced on your clay colloids and your humus colloids. But we also need the decay to be right. Now, in that rhizosphere, this is why I call it the plant stomach. The majority of the bacteria is found either in the rhizoplane or the rhizosphere, which means it's immediately adjacent to the roots. Why? One of the gentlemen here asked a question about organic matter decomposition reducing with changes in chemistry. So if our pH is really high, most bacteria want a pH around 7.4 or I believe 7.4 to 7.0 for roots and bacteria. They want that pH there. Now, this is not bulk pH, okay? When you take your soil to a lab and you have it measured, that's bulk pH. They come back and they tell you the pH of your soil is 6.5 or 6 or 5 or whatever it is. That's bulk pH. We're talking about microenvironment pH of a root or a bacterial organism. That is the pH immediately around that organism, protecting that organism and balancing chemical processes inside those cells. That's the pH that needs to be 7 to 7.4. And that pH is manipulated by exudates. That means things that organisms push out of their body to manipulate that pH just in that little environment. So, plants will also push hydrogen ions out of their root cells to manipulate the pH in the rhizosphere and in the rhizoplane. So when plants do this, bacterial organisms will move there because they don't have to expel energy to produce that same pH, to manipulate that pH. So if you can think of it like this, how much energy does it take for you to stand outside in the cold and shiver and shake and keep yourself warm versus just coming inside this room? Right? If you had to think about how many calories your body would metabolize being outside in the cold versus just hanging out inside by the fire, you wouldn't have to eat so much, but that doesn't keep us from eating a lot, but we don't have to, we don't have to utilize that energy. It's the same for bacteria. They want to be where the environment is going to be most suitable for them. And if that environment is going to be, it's going to have a pH closer to what their desired pH is, then it doesn't have to metabolize as much carbon. So when you let me rephrase that. When you balance your soil chemistry, you will usually balance the pH. It'll fix itself, usually. There are some exceptions. There's a few soils out there that are real crazy, but for most soils, you'll correct that pH. When you correct that pH, microorganisms no longer have to metabolize as much organic matter, which means energy, to manipulate their environment so that they can survive. You guys got that? And if you're not metabolizing as much organic matter, so that you can manipulate your environment, so that you can be comfortable, then your organic matter will actually build up in your soil, which means that you will see increases in that percentage of organic matter, both biologically active, intermediately active, and stable organic matter fractions. All three of those fractions of the organic matter will stabilize, and they will begin to build up if you can get that right. So you can see just off the top of your, without doing a whole lot of, or without having a whole lot of understanding of the sciences, why pH is given so much respect. Unfortunately, how to get it right is usually not given that much respect. They turn to lime, 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 lime some more, or other things, but they don't get it right. Oftentimes, they also turn to ammonium-based fertilizers. Ammonia, which is NH4 positive, will quickly be metabolized, which is called through ammonification, to be formed into nitrate. When it does this, it releases 2 hydrogen ions for every single element of ammonia. So what does that do to your soil? It acidifies your soil. It doesn't matter the form of ammonia. Plants will only take 2 types of nitrogen. They only like it in 2 forms. Either they want it in ammonium or they want it in nitrate. Nitrate is NO3 negative. Ammonia is NH4 positive. If the plant is taking up a lot of ammonia, it will convert that ammonia in its plant cells, and it will dump that hydrogen out of its roots, further acidifying the soil. So the types of nitrogen that you're using and you're amending your soil with, if you decide that you want to use those synthetic forms, is going to affect your pH. But the problem is that most people don't understand how it's affecting their pH, and it's how a lot of times you'll lime, and then you'll come in and you'll double down. A lot of farmers will double down on nitrogen because they don't want to lose their crop. And then they can't seem to fix their pH. They blame it on the rain. They blame it on the soil. They blame it on whatever they want to blame it on. The pH will not be corrected. And then on top of that, they come in and they put in a whole bunch of synthetic phosphorus, fertilizers like monoammonium phosphate. That phosphate will immediately react with that lime they just applied to form calcium phosphate, which is also known as apatite. That, again, does what? It locks up your calcium, it locks up your phosphorus, and it lowers your pH. How? By chemistry processes. It's all in understanding the chemistry. You make your calcium, it won't lower the pH. It won't change the pH at all. It usually ups the pH, but your calcium availability when you go and you have your soil tested will not change. And you'll find that you don't have any calcium in your colloids. Why? Because it's tied up with your phosphorus. Because you're adding synthetic forms of phosphorus. This is where people get into serious trouble adding too much phosphorus. In one of the fields in Massachusetts that we've had 5 college farms purchased, the previous farmer, it was leased to somebody else, but he grew potatoes. And he grew nothing but potatoes every year, and he'd been doing it for many years. When I had that soil tested, pH was 4.5 or 5 or something like that. It had 6,000 parts per million of phosphorus. It was insanely high. And you would lime that field, and it seemed like it did absolutely nothing to change the pH. And it's why sometimes folks say, hey, I lined my field and the pH just won't change. And they don't understand why. They said, look, I did all the Albrecht soil balancing math that you're supposed to do. I went in there and I figured out what my CEC was. I figured out, hey, I need this much lime to change it. They make their applications of lime. They come back to say, hey, my pH didn't work. This Albrecht nonsense doesn't work. I don't want to practice this. But what they're not understanding is, where is that calcium going? You got to understand where that calcium is going. And a lot of times it's tying up with other things that should have never been applied. And in a lot of farms, like this particular farm, you'll find a lot of literature that says potatoes love phosphorus. So you better double or triple down on phosphorus. You do that for 10, 15 years straight, you're going to have a lot of phosphorus in that soil. What ends up happening is that phosphorus will bond with not only calcium, but it'll bond with magnesium, it'll bond with arsenic, it'll bond with copper and zinc, and it will form precipitates, which are solids that it cannot break down through standard metabolic processes. Once this happens, it's called supersaturated. Your soil becomes supersaturated with phosphorus, and phosphorus, a nutrient that is not supposed to move more than a couple millimeters in a lifetime, in our lifetime even, It's not supposed to move through the soil profile at all. It's not mobile. But it becomes mobile when your soil is super saturated with phosphorus. And what that does is it starts producing eutrophication. What is eutrophication? That's when you get a bunch of phosphorus in your rivers, your streams, and your water tables. And all of a sudden you have these algae blooms that take off. So by the time you get to the point that you're causing eutrophication, you've already seriously messed up your soil. And this has happened all over the country, especially in the Midwest, especially in the upper Mississippi River Delta. A lot of this is happening out there. So this is where some of that stuff goes. Now, I'm going to take you, we're going to keep talking about metabolic processes of organic matter. We're looking here at arbuscular mycorrhizae and ectomycorrhizae, also known as endo or exomycorrhizae. Endo, indoors, inside. Ecto, exit, outside. All that means is you have two different types of mycorrhizae that form symbiotic relationships with your roots. Some of them are internal to the root. They come inside the root, as you can see to your left here, and they will actually form hostoriums inside of the plant cells. This is very similar to the way Phytophthora infestans works and many other funguses that are actually pathogenic, that kill your crops. But this one is not pathogenic. This is symbiotic. What that means is that it uses pretty much a lot of the same tools, but it doesn't seek to kill the plant. What it wants to do is actually form a relationship and share things. Exomycorrhizae, you see to your right, will form around the root system and protecting the root system mostly. What is interesting is the one to the right, ectomycorrhizae, dominates forest floors, while the one to your left is usually in your grasslands, prairie lands, and other places. So you're more likely to find it in arbuscular mycorrhizae in your garden or your agricultural lands. And if you've got an orchard, you really want to develop the ectomycorrhizae fungus, and you want to get those species down. And the reason why is because a lot of research has happened, even just in the last couple of years, that has shown that mycorrhizae doesn't just reach out and expand your soil rhizosphere. Now, the rhizosphere, like I was sharing with you guys, is that area right around your root caps, right? And when a plant starts putting roots out, it's so small. I don't remember, it was 0.01 or 0.001% of the overall volume in the soil. In other words, the surface area is really tiny. To maximize that surface area, you need things like fungus. Now, why do you want to maximize your surface areas? Because if you increase your surface area, you increase diffusion, and you increase the rate at which nutrients will enter into the fungus or enter into the plant. In other words, you're increasing the amount of area that your nutrients will be able to exchange, or I'm sorry, that your plant will be able to exchange nutrients with the soil. So that's where mycorrhizae fungus really gets popular and has earned a lot of its respect. However, there's more than just that. A lot of research has found with perennials in forest, as well as in orchards. I think they tried it in hazelnut orchards and peach and pear orchards in Washington. And what they have figured out is that when these fungal networks form in the soil, they become like communication networks. And when you look at an orchard, they all begin to interconnect because fungal organisms are huge. They are huge, and they form hyphae, and they just go everywhere. And a hyphae is like a root system, like what you see sticking out on both sides. These are hyphae. I think it says that, but this is a low-quality picture. You probably can't make that out. They start to form these hyphae that go everywhere, and they begin getting into all these different roots, and they start going down into the parent material in that soil, and they start weathering that material, accessing nutrients that the plant could never access. But they also, which is very, very crazy, they started looking to see, hey, if let's just say we take these 2 or 3 plants over here in this orchard, and we put excessive amounts of nutrients there, and we don't put any at the far other extreme of the orchard, what would happen? And they started taking tissue analysis and making these additions and deductions in their amendments, and they put isotopic nitrogen in there to figure out what it was doing. And what they discovered is that nitrogen that was poured almost 300 yards away was found way out in a different orchard when they tested the leaf tissue. And what they discovered is that the fungus was actually sending nutrients to other plants and vice versa, so that they were sharing excesses and deficiencies to balance each other out. This is crazy. This is relatively new. I mean, absolutely just baffled the whole scientific community. They never thought that they understood symbiotic. They understood, hey, you know what? I'll give you $1 and you give me, a burger or something. But they never understood, hey, look, I have all this excess. Share it with, your distant family somewhere. They never thought that it would do something like that. I mean, this type of giving, you don't find outside of the Scriptures. This type of caring, you don't find outside of the Scriptures. But then they took it another step. They took it another step further, and they said, hey, look, now, this time they did these tests in a forest, and they were looking at Douglas fir, a type of a conifer tree that is grown particularly for lumber in the Pacific Northwest. And they wanted to know if mycorrhizae and the host plant would be able to actually recognize its own saplings. In other words, it drops its acorns down and it drops its seeds down around that forest floor, and some of them will take off. Now, if you look at a forest, you notice you have these huge tall trees. If you spend any time in the forest, in the Pacific Northwest, there's tons of forest. And if you go and you walk in the forest, you see these trees, some of them almost 200 feet up in the sky, and you say, the sun never gets down here. How do these trees, so small, get the nutrients, especially the photosynthase they need, to eventually make it 200 feet up into the air? And what they discovered is that when those saplings take off, that mycorrhizae will actually form associations with the roots of those saplings and start to feed its own offspring, its own progeny. Isn't that crazy? I mean, they can actually recognize the genetics of other species in that forest. And when they went and they took same Douglas fir, but from a whole other part, I think they brought it from Canada or somewhere else, so the genetics were, essentially it's still a Douglas fir, but it's a different, it's a far distant cousin, and they brought it in, it favored its offspring over the one that had been brought in from a different forest, even though they were the same species. That was crazy. That was another thing they never thought that would happen. So then they going back to orchards now. The reason why I bring this up is because it is very, very important that with orchards and any of your perennial crop that you really try to get those fungal communities developed in your orchard. Why? Because it will balance. Because here's another one. I want to tell you, I'll share another one with you, another trial they did. They actually inoculated, and I don't remember if it was fire blight or if it was, I can't remember which type of blight they inoculated the plants with. But they started, or maybe it was pest. Man, my memory is, I can't remember if it was a fungal pathogen or if it was an insect. And I want to say it was an insect. And I'm thinking that it was actually aphids that they put on one plant. Now, plants have the ability, when they sense something, and I'll talk a little bit about it a little bit later, but when they sense that there's certain attacks coming in, so in other words, their leaves, as soon as an organism gets onto a leaf and it begins to actually attack the cells have within them the capacity to sense certain phytoalexins and things that are actually released into that plant cell by the pathogen or the pest. And it can signal to the rest of the plant, hey, it's time to build up our, what is referred to as system of defenses, plant system of defenses. And it's essentially the immune system of a plant. And it starts building this immune system, so now it starts to actually release things to fight that pathogen or that pest. But what they never thought would have happened is that if they did it with one tree in the orchard, that every single tree in the orchard would also activate its systemic system of defenses. And this is only made possible through mycorrhizae. So it was very interesting that with pests and pathogens that it would actually communicate with other plants and say, hey, I'm being attacked by aphids, develop start expressing a certain part of the genome that would actually give it resistance to that pathogen, some type of resistance to that pathogen. It's really interesting, but certain genes are not expressed unless they're called for. And that's the science. It's the same with us. Certain genes are not expressed unless they're called for. But you got to have the nutrients to give that. So this is, again, more talk about mycorrhizae. So here we're going to go back to organic inputs. Now, aggregates, soil aggregates. We're going to go back into the soil. We're looking at soil aggregation. We're looking at soil clods. How does this thing build up? How does, soil is really just a whole bunch of weathered minerals. Whether it's clay, humus, or silt, it's just weathered minerals. How are all these things held together? What holds them together? What glues them together and makes that soil aggregate that we see in the soil? And we say, hey, this is good soil versus, hey, this soil is dead. Good luck growing anything there. And what that is going to be your exudates, whether they're secretions, mucilages, mucigel, and lysates is what they're called. And these are all different carbon substances that come out of roots and out of microorganisms to actually glue and stick this soil together. And when you're constantly pounding the soil with a plow at all the wrong times of the year, and you're not plowing intelligently, I'll talk a little bit more about that. I don't believe in, people always ask the question, to till or not to till? And they come up and ask me, should I till or should I not to till? The farmers ponder, right? To till or not to till? That's at least the 21st century's farmer. And my answer to that is intelligent tillage. I'm going to steal this one from Whitmar. That's who it was. God plowed Pharaoh's heart and he hardened it, is what he said. I don't know how many times he said that. But essentially the principle is, there needs to be some plowing, some breaking of our heart. But if the Holy Spirit doesn't come in, what good is it, right? It serves us no good. If we're going to plow the soil, you better be doing it for a good reason. You better have a purpose, and you better be adding something to it. You don't just go and plow the soil because it's March. I'll talk a little bit more about microbial metabolism, and we'll look at some of that in a second. This is a chart looking at water effects. We're going to look at the water effects of microbial metabolism. So what is that? I'm going to keep saying that word, and I want to make sure that everybody in this room understands what microbial metabolism means. And that means organisms of any type, particularly bacteria, actinomycetes, and fungus, breaking down organic matter, metabolizing it. That means breaking down those carbon structures, those phenolic compounds, breaking down the chitin, the lignin, and these other things, and releasing the nutrients like the proteins, taking those proteins down, breaking that amine group off of it, taking that amine, which is the NH2, and turning it into NH3, eventually converting it to NO3, which is nitrate, making it available to the plant. So how do you get a protein from a protein to a nitrate so that your plant can use it? Is a very intelligent intelligent process that, to me, only by design. But it's strictly microbial. This picture shows the effects of water. When the soil is desiccated, which is A, which means that there's some moisture in there, you'll never get all the moisture out again unless it's very, very hot and you put it in an oven and you heat it to 500 degrees Celsius. If you don't do that, you're not going to get all the moisture out of the soil. So you have some moisture in there. In those small micropores in the soil aggregates, you will have bacteria and you will have other soil microorganisms inside of there. The moment you begin to add moisture to it, they can finally move around. Once they move around, it's really, the soil is like the Amazon. I'm sorry, not the Amazon, the Sahara. You have lions and you have cheetahs and you have gazelles and you have all this other stuff. If you could just take that, all those images you have in your head when you think of National Geographic or Discovery Channel, and lions and tigers and all these things running around and chasing, water buffaloes and things like that, that's exactly what the soil is. Except you have all these different organisms that are chasing bacteria and protozoas and worms, and they just running around killing each other and eating each other. And every time they kill somebody, there's a whole bunch of nutrients released that are not needed. In other words, there's a lot of waste, but that waste is consumed by plants. That's why they say microorganisms eat first and then the plants. That's why they say feed your soil. That's what's going on inside the soil. When the soil is saturated, supersaturated, that means you have 100% of its water potential. Well, it won't be 100% water potential because the soil, that would mean a glass of water, but 100% of the pore space is consumed with water, or full of water. There's no air in that soil. Microorganisms can now move through that soil. Through diffusion, nutrients will move, organisms will move, and things will just get around that soil so fast, and it happens quick. Diffusion is, milliseconds. You're talking about an element of magnesium moving from, one particle to a different particle is something that happens very quickly. But it happens with saturations in soils. There has to be, God made by design, these fluctuations of moisture and lack of moisture, and then you're saturated, you're flooding, and then you're drying, and then you're somewhere in between. Because when these processes are happening, there's differences in the way that microorganisms move around, how they metabolize organic matter, and what organisms get to die and what organisms get to live. So that makes a difference of when the lion catches the gazelle and when the lion doesn't catch the gazelle in the soil. It's very interesting how this moves. Then you also have changes in gas transport. We need oxygen going down there. You need you need carbon monoxide, you need all these different gases, especially nitrogen. 78% of the air is nitrogen. If we're going to fix rhizobium, that species of bacteria that fixes nitrogen so well, is going to actually fix nitrogen, you need gas exchange. You need that nitrogen gas to get into that soil in order for it to do it, right? Whatever gas was put in there when you tilled that soil is going to be long gone just a few days after you tilled it. So how does it continue to fix nitrogen? There has to be gas exchanges. So this is why you don't want to have it fully saturated with water, because there's hardly no gas exchange. If you look at the chart there in the middle in B, I don't have a pointer stick, but over here you see gas transport and then volumetric soil moisture. The more moisture you have in the water, the lower your gas exchange. Your solute transport will move. Metabolic costs come down. In other words, it requires less energy with more moisture and more energy with less moisture for microorganisms to move through the soil. Predation, it's just like above ground predation. This is soil below ground predation of predatory species attacking other species that are usually herbivore or some other heterotrophic bacteria. Will actually increase as that water volumetric soil moisture increases. This is a slide that you could stare at for hours here and just think about and ponder the differences in the behavior of microorganisms within the soil based strictly on water effects, nothing else. How much water is in that soil? Here's another one, fungal bacteria ratios based off of pH. What does pH have to do with our fungus to bacteria ratios? And what we have found essentially is that the more acidic a soil is, the more favorable the environment to fungal pathogens. So I talked about the genetic diversity of different species in a soil. And this is one of the things where pH, just like moisture, at the bottom of a pond, you're going to have all the same genetics that you have at the top of a hill. It's just that there's going to be a lot more anaerobic bacteria at the bottom of a pond than there will be at the top of a hill. You understand the difference? It's not that it's not there. It's there. It's all around us. It's just that it's the environment for the anaerobic bacteria is favorable at the bottom of a pond versus at the top of a hill. It doesn't mean those species aren't there or that they can't get there. It's a difference in moisture. Now we're looking at a difference of pH. Okay, you got both these charts here, as well as carbon to nitrogen ratios is another fungal to bacteria, or another characteristic that will dictate whether the soil is predominantly fungal or bacterial. And you see the chart here showing if your carbon is very high, you're going to favor fungal organisms. Why? Because fungal organisms are the only ones that can break down chitin. Bacteria can't break it down. If you look at these complex carbon structures and the alpha and beta bondages that they have with the phenolic compounds, it takes certain enzymes to break those down. Bacteria is not advanced, or if you want to say advanced enough, they say it hasn't evolved enough, but whatever. I don't believe all that. But anyway, it's not as complex, and it doesn't make those exudates to actually break down those bonds and break down those high carbon plant tissues that you would find in, say, a wood chip pile. So your what was that? I forget. Back to Eden, thinking it's not going to work there. It's going to tie up so much of your nitrogen. It never fails. Here's some modeling for temperature effects. Temperature is another variable that really, really, really dictates when to plow. Why? Why? Again, I say intelligent tillage. When should you plow? When should you not plow? The temperature here is in Celsius. And the rate of metabolism here is it's on the y-axis here. So Arrhenius, Lloyd, and Taylor from way back in the late 1800s, early 1900s came up with these two theories of a rate of reaction. In other words, they're trying to make a mathematical equation for the rate that microorganisms will metabolize organic matter. Now, mind you, microorganisms don't do math. It's the first thing you got to think about. They don't do math. These are just equations to try to give you an idea of the metabolic rates of these organisms with respect to temperature. So these were the first that came up, and we see that they're relatively the same, but it won't go forever. At some point, it's going to reach a temperature which is really almost about 30 degrees Celsius, which is about 85 degrees Fahrenheit. where those equations are no longer valid, and then we went to the macromolecular rate theory, and this is actually much more valid. It looks kind of like a bell curve, and with its peak right around 90 degrees Celsius. And this is soil temperature, not environment. Okay, this is not ambient temperature. This is soil temperature that we're looking at here. So when the soil temperature is about 90 degrees Fahrenheit, which is 35 degrees or so, maybe 32, 33 degrees Celsius, that's when you will have your highest rate of microbial metabolism. That's when organic matter will break down the fastest. And that's the point where nutrients will be made most readily available to plants. That's really the only important thing you need to get out of this slide. You don't need to understand the math and all the graphs, but what you do need to understand is at what temperature would the maximum amount of nutrients be available to the crop. And you need to line those up with the point, particularly nitrogen. You need to line that up with the point as farmers. We need to line that up, if we could superimpose on this the graph of when plants want the most nitrogen. And if you can line those up, you'll have the nitrogen you want at the time you need it for the crop you're growing. No, it's actually way before fruiting. Yes, by fruiting, you're pretty much all done sucking up nitrogen. So here's a rhizosphere effect. Now what we're looking at is the distance from the root surface and the concentration of microorganisms. This slide probably should have been, I think I jumped the gun when I started talking about this. But this is a factor of 10, folks. This is 1 to the power of 10, what is changing here. So you're seeing some pretty big, fungus, streptomycetes, and bacteria right there, 0 millimeters away from the root, or pretty much essentially at the root, which would put you at the rhizoplane, not the rhizosphere. But this is it says rhizosphere because as soon as you get away from that, you're in the rhizosphere. But you only need to get 10 millimeters away from the rhizosphere, and you see a huge sharp drop on each category: bacteria, streptomycetes, and fungus. Their actual population, their density levels goes down the further you get away from the root, which again goes back to the theory that they want to be adjacent to the root, where they will consume less energy to manipulate their environment. This is other rhizosphere's characteristics. We're looking at water uptake based off of distance from the root. So you water the entire field, right? But how much of it is actually being used? Okay, so if we turn the sprinklers on, how much of that water is going to actually get used by the plants? Well, when you turn a sprinkler on, depending on, if you're in the West, where humidity likes to hang around at 10 or 5 sometimes, and real low humidity, you lose as much as 50% of your water before that water droplet ever hits the ground. That's a lot of water to lose. And then once it gets to the ground, you're watering the entire field, but you've got, say, a row crop like corn with a 30-inch spacing. What does that mean for water consumption? So this is where Netafilm, the corporation that makes irrigation systems, came out with underground drip tape for corn, and they've seen reductions compared to sprinkler irrigations. I think they're consuming, if I remember right, it's either 10% or 25%, somewhere around there, a quarter to a tenth of the amount of water that was necessary for sprinkler irrigation. And that means a lot when you're out west. And everybody here is from all over the country, but when you're out west, water is important. So we start looking now, okay, what is the water? Remember we talked about transpiration, the sucking of the plant. It's like a straw, sucking moisture from the soil through the plant all the way out the leaf, right? Bringing nutrients in with it. Now, where's the water disappearing and how far can that straw really suck? And this is what this graph shows. When you're 1 millimeter away from the soil, you pretty much can move almost all your water out of there. And as you start to get to about 2 centimeters, you've lost most of that, that negative pressure. In other words, 2 millimeters away from the soil, from the root hair, you're already losing, your water consumption drops significantly. It's rather interesting. And the further you get away from it, the greater the effect. But pretty much, by looking at this graph, you can say within the first 1 to 2 millimeters of that root hair, that's where the majority of your water will be pulled from. So it gives you an understanding of really where you need to be putting your water. These are compositions of exudates. We're looking at sugars, amino acids, organic acids, etc. There's actually, there's some people that have talked about fertilizing with sugar. Sounds crazy, huh? But fertilizing with sugar. Because the compounds will, they'll cause, essentially it's a substrate. So your bacterias that consume, that will actually survive, and mostly your bacillus, your lactobacillus, you can actually fertilize with, with dairy if you want to. You can go get the old milk that they don't sell at the dairies and actually dump it on your fields. And what that's going to do is going to drastically multiply your lactobacillus genus. Anything in the lactobacillus is going to just take off. And those are actually a lot of your anaerobic species that will break down organic matter at some of your lower depths where your air is not going in. I think it's been most effective in some of the clay soils where it's real hard to get gas exchange and getting oxygen down there for some of this bacteria down there. But anyhow, these are some of the exudates that plant put out. So abscisic acid, that's a really interesting one. Abscisic acid has been used to reduce blossom end rot. Gibberellic acid is actually, both of these are plant hormones, abscisic acid and gibberellic acid. You also have catechol, which has a lot of things bonded to it, but these are all carbon, again, I said the science. This is all organic chemistry that you got to understand here to understand what is being broken down and how these things break down and how they work in the soil. Let's see, spatial extent of exudates. Okay, so we're looking at, again, 2 millimeters seems to be the rhizosphere. 2 millimeters, by the time you hit 2 millimeters away from your roots, your plant exudates have severely dropped off. So if your plants are going to be putting anything into the soil to manipulate their environment, really most of it is done within the first millimeter. And in this example, the exudates really don't get far. They really don't move. They stay pretty close to the roots. Here's another one. I told you that roots can actually manipulate the pH. So here we're looking at what they did here. These three images are the same exact photo, the same exact thing using different technology. So we see here a container. I don't remember. I want to say it was clover, maybe alfalfa, but I don't remember. It was a legume here. You see all the root hairs through here. Now, how does the pH change? How does it vary? Remember, I told you when you take your soil to get sampled and tested, they give you bulk pH. What we're looking at here is the pH at the actual rhizosphere and the rhizal plane. And you can see the changes and the enzymes that are released. And if you could put that image on top of the other image here, you notice that those changes where the most activity is right there adjacent to the roots, relatively close. So when your soil really varies significantly and what's going on based off of the roots, the exudates and microorganisms that are working with it. I think this is going to be our last slide here. Rhizosphere dynamics. We're looking again at the fungal hyphae. So the rhizosphere is where you want your fungus to actually make its physical connection with the plant. But those hyphae will take off and go all over the place and collect nutrients that will ultimately bring to the roots. And this one here we're looking at an amoeba and how it's, microorganism will stay adjacent to the plant. Organic matter will break down. So this is just, these pictures of organic matter is just, organic matter comes in so many shapes, sizes, and colors that they just use generic images to represent it. But this is just organic matter breaking down by microorganisms and releasing some of these nutrients. But you see the same thing down here with fungal hyphae will bring a lot of these nutrients into the roots as well. I believe that is going to be it. Yes, we are. Okay. So this is going to conclude this particular talk here. I think that I covered quite a bit of things here. I know some of this stuff is pretty heavy. I'm trying to run through it and use basic terms, but I really got to understand some of these things if you're really going to be profitable at what you're doing. You don't have to understand all of it, I don't believe, but you got to understand some of it, especially with water management and what roles you can expect microorganisms to play and how pH affects the soil. Questions? Boy, we had a lot. Okay. I'm sorry. I think it was the lady back here with the glasses.
Okay. You mentioned that the pH will influence which microbes are favored, if it's fungal or bacterial, and I somehow didn't get...
Okay. If a soil is predominantly acidic, it will be mostly, and I speak bulk soil pH, okay? If the bulk soil pH is predominantly acidic, it will favor fungal organisms. When you start, if it's mostly alkalinic, there'll still be some fungus, but not much. It's mostly bacterial and actomycetes.
mycorrhiza, where do they fit in, the mycorrhizal?
Okay, the question is, where does mycorrhizae fit in? Mycorrhizae fits in that it connects with roots and it goes everywhere. Mycorrhizae is a type of fungus. It comes in globular mycetes, actomycetes, basidiomycetes, a lot of different categories of fungus that are out there. But mycorrhizae and many other fungus will form symbiotic relationships with other plants and other organisms like lichens and things like that, and they'll spread out and they'll go down into the bedrock. Well, depending on how deep your soil is and what's there, they'll go into the rocks. They'll go into with other plants. It's a huge organism that is completely underground, and it really connects with almost everything down there. So where it fits in is a very complex question to ask.
I just meant which pH favors it.
Oh, acidic. Well, that's depends on species, actually. I'm sorry. That one depends on what species of mycorrhiza you're looking at and what categories it falls under, because there's a lot of different types of mycorrhizae. They're not all the same. I'm sorry. I think this...
For greenhouse growing, you need to pay, I guess, close attention to your water saturation in the soil, because you tend to run dry.
Yeah.
So are you saying, based on the earlier slides, that having a variation in saturation is important for gas exchange and nutrient exchange?
Yeah. Okay, so a lot of the greenhouses that they use in the Netherlands and in other places will use soil moisture. These are really advanced, real high-end, fancy. They got a lot of equipment. It's not something you'll ever use, but what they try to do is they try to manipulate. They based off of soil tension, and they're constantly changing things because what it does, like I say, it favors different environments based off of what they're trying to accomplish. If you're in a more simple, which is what most everybody in here is going to have, is just a simple greenhouse. Most of the time we challenge as growers, as farmers, we have a challenge of keeping the soil moist, not keeping it dry. Well, it depends on where you. Well, I mean, even in Virginia with plenty of rain, we struggled, and even over in Massachusetts, we struggle sometimes to keep the ground wet because it's a greenhouse and it never sees rain. There's some losses to never seeing rain. Rain is acidic. That's where a lot of the hydrogen ions came to begin with, to wash that soil out and remove those nutrients and get it acidic. But when you're irrigating, you really need to be keeping your moisture at a desired level, and I would recommend using some type of simple moisture probe to have in these houses and base your watering off of that, as opposed to just saying, well, it's 9 o'clock and it's Tuesday, let's water, and you really don't know if you're getting enough water down to your crop. So water management is a complex question that based off of soils, environments, and what you're growing in. I don't have a basic, easy answer for that.
Does mycorrhizal affect water transport or exudate transport?
I don't, well, from what I understand and how I understand the science, mycorrhizae has the capacity to move a lot of things more than just simple cations or nutrients. It can move different plant hormones around, and so I would definitely, my answer to that question would be a definite yes. Christine?
Yeah, you started to say something that the soil temperatures for tilling, and then you went into the soil temperature for the highest rate of microbial activity, which was
okay.
around 90 degrees. So what was your point on soil temperatures for tilling?
Yes. Okay, the question here is, if the soil is 0 degrees Celsius, that means the soil is maybe just barely thawed out if you're going to go out there and plow it. So if you were to go out and plow a soil, the argument is with plowing is that you're destroying environments, you reduce your organic matter because the microorganisms, well, because you introduce a lot of oxygen, and the aerobic bacteria will begin to break down and accelerate the metabolic process of that organic matter, releasing a lot of nutrients, which adds to leaching and getting nitrates, particularly in your water tables, and many other unfavorable processes. So if you're going to till intelligently, you really want to till when the soil is not as warm, because what's going to happen is any oxygen or any other things you introduce would have been slowed down. In other words, you won't have that acceleration in microbial metabolism that you see when you till a soil in warmer temperatures, from say 25 degrees Celsius all the way up to maybe 40 degrees Celsius. is when you would see the highest rate of microbial metabolism. So I would encourage, one of the things that I say is that you should, if you're going to till for the purpose especially of adding nutrients, which if I was talking about cation balancing, which I'm not, but essentially if I was going to do that, that was my objective. My soil is a mess. I want to get it right. I want to get it right away. I use rotary spading, deep tillage in the spring when the soil is still relatively cold. Its metabolic processes are low. It'll semi-compact again, but during that time, I know that I have those nutrients. I know they're in the soil, and I know they're mixed in thoroughly so that I can expect those changes that I want to see in the colloids by making those additional amendments.
So
Next.
what was the temperature? Could you say?
Oh, well, you're not ever really going to be out tilling at 60 degrees Celsius, but it would be pretty much from just barely thawed out, like probably what the soil is outside right now here in Texas, all the way up to about 20 degrees Celsius. Okay. It's about 70, 65, 70, soil temperature. Again, guys, this is soil temperature, not the weatherman's temperature. This is soil temperature that's important. Okay. 68? Yeah, about 68, that's right.